Welcome to your Unit 4 exam review. And you made it to the last exam. And with Unit 4, it's just over two chapters 22 and 23, covering digestive system and metabolism. And like always, you have an exam review located in the Dr. Higgs tips section. And when you go to Dr. Higgs tips section and you click on your Unit 4 exam review, it brings up a little Word document that looks like this here. Make it a little bigger. And it's just like all the previous exam reviews I've written. It's just me rewording the objectives you see in Blackboard. Hopefully it makes it a little bit more straightforward. And like always, go to bullet point by bullet point, making sure you know it in the detail it was taught to you. And towards the end of this exam review, I put in a little extra detail. Hopefully it will help with chapter 23. Chapter 23 over metabolism, it's kind of tells you in the name, it's metabolism. So we're really talking about breaking things, building things up. And when it's dealing with your body, it's talking about the food you eat. All right, all your food sources, your food stuffs are going to be broken down to be used for energy and to build things up in your own body. And when we look at breaking things down in the human body, we keep it simple. We look at things like carbohydrates and sugars. Remember, sugars like glucose can be used to build large glycogen complexes, which are carbs. So when you talk about glucose catabolism, yes, you're talking about sugar, but you're also talking about carbs. The same way you break down a teaspoon of sugar is pretty much the same way you'll break down a slice of bread, because that bread is carbs, and chemically, those carbs, when they get broken down, they will get broken down into their individual sugars. But this detail is just kind of giving you the steps that you need to know. I know these metabolism reactions are lots of reactions occurring on. When we talk about things like glycolysis and the citric acid cycle, sometimes called the Krebs cycle, these are a series of chemical reactions. No, you do not need to know the series of chemical reactions. This little document kind of gives you the key highlights. What you really need to focus on for this class is just what is the starting material, what are we breaking down, and then what do we end up with. All right. Breaks everything down. When we talk about breaking down sugars, it starts off with glycolysis, then you'll go on to pyruvate phosphorylation, and then the citric acid cycle, and you end with electron transport chain and chemiosmosis. And it tells you all the things you're starting off with in each step and everything you'll end up with after each step. All right. And so I do that for glucose, for fats, and for amino acids and proteins. All right. So I'm not going to go into a lot of this detail because I've written it out for you. But as we go through the review, like usual, going through every single bullet point, I'm going to focus on ones I think you might need a little more help with. All right. And yes, like usual, I'll be using your lecture PowerPoints. Okay, so chapter 22 and chapter 23 lecture PowerPoints are good enough, and I'm going to be using them to help walk through this exam review. And we're going to start off with chapter 22, the digestive system. One of the things you need to know is very straightforward. What are the basic functions of the digestive system? And like always, before we get started, you know this already. You just don't know the fancy names we give to stuff. Remember, we're talking about digestive system. You know at least two things way back from chapter 1, way back from when we talked about organ systems, way back to when we talked about the digestive system. Remember, you had to know an organ or two and a major function. You know at least one organ. It's things like your stomach or your intestines. And if you know stomach and intestines, you know at least one major function is to break down food for nutrients. This system, as one of its functions, helps you to break down food and nutrients for the vitamins, minerals, and other nutrients you need in that food. And by now, all went through kindergarten, so we all know everybody poops. So we should know it eventually. Not everything gets absorbed. There will be stuff left over, stuff your body doesn't need, and you'll get rid of it. We call that excretion. Everybody poops. You call it pooping, but remember, In anatomy, we're fancy. We call it excretion. So you knew some functions already before we even got started. Again, you know these functions of the digestive system. You just know the fancy names. For example, for me to break down food for nutrients, I need to first take the food in. I need to eat the food. I need to bring the food into my digestive system, called ingestion. That's one function, just ingestion. That's just you bringing food into your body. And for most people, you do that via the mouth. Keep going. Other functions. Again, think about it. Think about eating a Whataburger cheeseburger. When I bite into that Whataburger cheeseburger, I'm ingesting the food. But then what's going to happen next? Well, you're going to chew the food. You're going to bite it, you're going to chew it up, and you're going to mix it with your spit. For you to mix it with the spit in your mouth, well, you needed to pump out the spit. You remember when your body pumps something out, we call that secretion. So you're going to pump out things like the saliva. You've seen this lecture PowerPoint, so you know there are things in saliva like enzymes, mucus. You're just pumping those things out of your glands in and around the mouth. You're just doing secretion because that's a function of this organ system. What else? Once you're done chewing it up, chewing up your food, mixing it with spit, well, you're going to swallow it and send it down the throat, into the esophagus, and eventually into your stomach. You're going to do a lot of movement of food. So one of the things you got to know in your exam review is how are you going to move food? Let me go bring up your exam review so you see which one I'm talking about. Here it is. Oh, it's the second bullet point. One of the points you need to know are what are the two major modes of motility. How are you going to move things throughout your digestive tract? Turns out your body uses two ways to do it. Overall, we call it propulsion. Overall, the process of moving food throughout your digestive system is propulsion. But there are two types of propulsion, two types of motility. One is a wave-like contraction of smooth muscle. So this rhythmic wave-like contractions of smooth muscle, we call it peristalsis. Think about being at a stadium and seeing people do the wave. And you see the wave, everyone stands up, their hands up, and then down, and you see that wave slowly approach. It's your turn. Well, that's kind of how peristalsis works. It's this wave of movement that pushes things through your digestive tract. But I mentioned it's not the only type of movement. Sorry. There's something called peristalsis, which I just explained, the rhythmic wave-like contractions of smooth muscle. But then there's something called segmentation. You see this a lot in your intestines. Segmentation is a mixing and churning of food. Think churning. If you have to churn butter and you're in the intestines, well, it's called segmentation. Think churning, like churning butter. So you could push things like contractions of smooth muscle, peristalsis, or you can mix and chew and churn things up. That's segmentation. And there are the two types of movements that we see primarily in the digestive system. But again, more functions besides just pushing things along, besides propulsion via peristalsis and segmentation, you also have digestion. Once that food hits your stomach needs to break the food down. Breaking food down is digestion. And turns out, like a lot of things in your body, you have more than one way to do it. One way to break foods down is physically break food into tinier pieces. You do it as simply as doing something like chewing. You bite into a Whataburger cheeseburger and you chew it up to mechanically break it into smaller pieces. Turns out your stomach could do mechanical digestion as well, because your stomach does a little segmentation. It could do some churning and pummeling of food. But you've seen your lecture PowerPoint already. You also know your stomach has lots of enzymes inside. Lots of enzymes that could also break food down. But you're using enzymes, you're using chemicals to do it. So we call it chemical digestion. Breaking food down, you're breaking it down so it's tiny enough to fit into your blood vessels, so that it's tiny enough to do another type of function, absorption. Absorption is just you taking your nutrients from your digestive tract and putting it into the bloodstream. That's it. But remember, we're not going to absorb everything. Everybody poops. Some stuff gets left out. There are some things that are not digestible and not usable by your body. So what are you going to do? You're going to get rid of it. It's called defecation. That's another function of your digestive system. Defecation is the process of this is just a fancy way of saying poop. So yeah, this chapter even talks about pooping. Oh yeah, in your exam review, you got to know a little bit about pooping. Oh boy, don't worry, we'll get there. Those are major functions, and we talked about motility through digestive tract. Again, just skipping through, hitting some highlighted areas. I think might need a little bit more explanation. Another thing, since we talked about movement, helps to know what's controlling the movement. Well, it's muscle that's doing the movement, yes, but it's smooth muscle, so you don't control it. You remember, there is part of your nervous system that can control or at least influence smooth and cardiac muscle. Remember, it's a part of your nervous system you don't control. It's called the autonomic nervous system, which is broken up into a parasympathetic and sympathetic division. Don't worry about that. Focus more on the enteric nervous system. It's your enteric nervous system that handles the majority of motility through the digestive tract. All right, and I believe I said that in your exam review. Know that the enteric nervous system provides control of secretion and motility through the digestive tract. All right. Keep going. You have to know some histology again. Oh boy, this is going to be like the 5th time you've seen this histology stuff. Don't worry. Remember, we went through this in your Lab Practical 2 review. If you want to go through your Lab Practical 2 review, just touch on the histology portion of your digestive tract. Go ahead and listen, and it'll help kind of clear some things. Otherwise, we'll look at this picture here. This picture is just showing a little section of the digestive tract, highlighting the 4 major layers you see throughout your digestive tract. Why I'm keep saying digestive tract. Remember, in this class, things have multiple names. You could call it the digestive tract, the GI tract, the gastrointestinal tract, gastrointestinal canal, or the alimentary canal. These are all synonyms. All right. I'm just used to saying digestive tract. But anywho, here we see a section of it, and it's highlighting the different layers we see. You got to know your 4 layers in order, either from innermost layer to outermost or backwards from outermost to innermost. The innermost layer is called the mucosa. Move a little further out, you get to a layer of tissue called the submucosa. Keep moving more superficially, you get to another layer called the muscularis externa. This is a large layer of muscle. And then the outermost layer is your serosa, sometimes called the adventitia, depending on where you are. And when we look at the mucosa, you might recall from your Lab Practical 2 review that it gets broken up into 3 sublayers. From innermost to outermost, the innermost layer is epithelial layer. So as we go through the digestive tract, when we talk about epithelial tissue in the, for example, has non-keratinized stratified squamous epithelium, while your stomach has simple columnar epithelium. We're talking about that epithelium layer of the mucosa. If you move a little further out, you'll see a little thin layer of connective tissue. And it's not cartilage, so it's possible to see blood vessels. This is called the lamina propria. And if you move out further still, you get a very tiny layer of smooth muscle. This is called the muscularis mucosa. And all 3 layers represent the mucosa. Keep going. Next layer I mentioned is a layer of connective tissue. Dense, irregular connective tissue is what we typically hear. So yes, you might be able to see blood vessels in this layer. This is the submucosa. Keep moving out. You'll get to the 3rd layer. This is a layer of muscle. Remember, it's typically smooth muscle, except for in the esophagus. where we'll see in the upper 1/3 of the esophagus is skeletal muscle, but as we approach the lower portions, you'll see more and more smooth muscle replace it. And everything after the esophagus will have smooth muscle as well. And then the outermost layer, again, some connective tissue with a little bit of epithelial tissue. This is the serosa or adventitia. It's holding all these other layers together. So this is just a little brief of the histology of the digestive system. It's 4 major layers and what tissues are in each layer. Then I mentioned some key features. I mentioned the esophagus has it has non-keratinized stratified squamous epithelium in its mucosa. And I mentioned in the upper 1/3 of the esophagus, you'll have skeletal muscle, the muscularis externa. In the middle 1/3 of the esophagus, you'll see a mixture of skeletal and smooth muscle. And in the lower 1/3 of the esophagus, you'll see strictly smooth muscle. All right. Remember, just check out your practical 2 review for some more details, logical features of all these parts. And then we can mention some of the organs. We'll be hitting these organs as we're talking about them. So there are things like your esophagus, your stomach, your intestines, small and large. All right. And plus the accessory structures like your liver, gallbladder, and pancreas. You don't know. We're just going to make our way now through the digestive tract, starting in the beginning and working our way to the end. So starting in the mouth and working our way through the anus and anal canal, and also those accessory organs, the liver, gallbladder, and pancreas. We make our way. You bite into a Whataburger cheese mentioned before. You're going to chew it up in your mouth. You're going to mix it with saliva until it becomes a softened mass we call a bolus, which you'll swallow. But before you swallow, you're going to have to chew it up, break it into tiny pieces, and mix it with saliva. Wow, so we got to talk about that. Where does the saliva come from? It comes from your salivary glands. You got to know your 3 major salivary glands. There's the parotid gland, located anterior and inferior to the ear, so kind of in front and lower down to the ear. Underneath the tongue, immediately below the tongue wall, you'll find the lingual gland, and lower down, kind of behind the mandible, you'll see those submandibular glands. All 3 glands are making components of saliva. And your saliva is more than just water. It's a mixture of other things embedded in the water. And remember, we talk about glands, but it's really the cells in the glands that's doing the secretions. So there's cells in your parotid, sublingual, and submandibular glands making more of the watery components and possibly some enzymes in your saliva. Call them serous cells, while there's cells making more mucus in your saliva. That's why your saliva is not like water. It's a little thicker, a little bit more viscous, a little tacky. It's because you have mucus in it, and it's coming from mucus cells. But we got to know what's really in saliva. Here we go. What are the components to the saliva, and what are the components doing? Well, for one, you begin chemical digestion with your saliva before you even get to the stomach. Turns out when you bite into that Whataburger cheeseburger, you already begin to break down the bun. Why? Because the bun is carbs. It's carbohydrates. Turns out you could break down carbs and starch with an enzyme called amylase. And this is coming from your saliva, from your salivary glands. So we call it salivary amylase. It helps you to break down carbs. What else? Turns out you could also just straight up kill bacteria with your saliva. You could kill bacteria because of an enzyme called lysozyme. So it turns out, when you were eating your Oreo cookies and you dropped a cookie on the floor, and because you're home and no one's looking, you picked it up and ate it anyway. You said 5-second rule. Turns out 5-second rule isn't real, but your lysozyme is. So maybe you didn't get because your lysozyme killed the bacteria. Keep going. What else? You have other things to help fight infections. You have antibodies, such as IgA, stands for immunoglobulin A. That's an antibody. And this is an antibody that's found in bodily fluids. And saliva is a type of bodily fluid. So you tend to find in your saliva. It's an antibody. And so, like a lot of antibodies, it helps to fight infections, pathogens, or infections. And even have bicarbonate ions. Oh, think way back to chemistry. Bicarbonate is a buffer. You remember buffers help like strong acids and bases. It's like you have your own antacid in your saliva. And it helps to explain something. If you ever wondered, oh, how's it that I can have heartburn in my chest? I feel that heartburn coming all the way up. It might even burn my throat, but when I bring it up into my mouth, it doesn't burn the inside of my mouth. If you ever had acid come up and it burned your throat, but not your mouth, now you know why. It's because you have saliva in your mouth. So as soon as that acid hit the spit in your mouth, it was neutralized. Oh, look at that. Who knew? Anatomy. So these are components of saliva. It's water. What things mixed in is the water, like lysozyme and IgA to fight infection pathogens. Things like salivary amylase to begin chemical digestion, as well as mucus that can also serve as a lubricant to help you when you swallow. Okay. So that's all that's referring to in your exam review. And I mentioned you have to know some sphincters throughout the body. I mentioned them in your lab practical 2 review. They're scattered because your text goes in order of organs, or really how food will travel to organs, starting with mouth through anus. So I'm just going to use this overall image of the digestive system kind of to help highlight. You got to know where these sphincters are. For example, you have a sphincter between the esophagus and the stomach. The sphincter between the esophagus and the stomach has multiple names. You could call it your cardiac sphincter, or your gastroesophageal sphincter, or your lower esophageal sphincter. You find it between the esophagus and the stomach. So if anything happens to this sphincter, esophageal sphincter or cardiac sphincter or lower esophageal sphincter, food might actually come up. You might have more heartburn, for example. Keep going. You have a sphincter between the stomach and the first part of your small intestines called the duodenum. The sphincter between the stomach and the duodenum is called the pyloric sphincter. Pyloric sphincter. Keep going. You have a sphincter between the last part of your small intestines called the ileum and the first part of your colon called the cecum. It's called the ileocecal valve or the ileocecal sphincter. It's between the ileum and the cecum. And then one of the last ones are between the anus and the external environment. Sphincter, and you have an external anal sphincter. They're both between the anus and the external environment. So think of them as the last stop, both the internal and external anal sphincters. So those are your major sphincters that we look at in this class. Again, keep going. We talked about the differences in muscle tissue in the different parts of the esophagus when we look at the muscularis externa layer. Remember, in the upper third, and smooth, and in the lower third, it's all smooth. Keep going. A lot of this is just things you need to know. For example, you need to know the part or regions of the stomach. Oh, yeah, images can come back on your lecture exam where we could show a picture of the stomach and point to an area of the stomach and ask you to name that region. Remember, there's several regions. There's something called the fundus, which is the top. The cardia is kind of like the entrance. Most of the stomach is the body. The pyloric antrum is kind of where it kind of tapers off. And the end is the pylorus, where you'll find the pyloric sphincter. Here it is all on this image. Towards the top, and the top of the stomach kind of tilts and faces your left shoulder. So here's the top up here. So that's fundus. Here I see the inch from where the esophagus comes. So this first opening area is the cardia. The vast majority of the stomach, this widened part, is the body. Then it begins to narrow at the pyloric antrum, and then you end with the pylorus and the pyloric sphincter. Those are parts of the stomach. Just gotta know them again. Then we look inside the stomach. Remember I mentioned the stomach could do chemical digestion. It could do chemical digestion because it has cells that make certain enzymes and hormones. So, like usual, you got to know it. You got to know your major cells in the stomach. What do those cells make? And the thing they make, you got to know what does it do. So, for example, chief cells make pepsinogen, which will be activated to pepsin, which helps to break down proteins. Okay, so let's bring that up. Still in stomach, we're just looking at the cells in the stomach, and we're talking about stomach makes with those cells. Remember, it's always the cells doing it, but it's really glands, clusters of cells. So we call them gastric glands, are what's making all these products of the stomach. But we know it's technically the cells in the glands making the products. So let's go through these major cells and see what they make. And like usual, their names, some of them help us out. Like this first one, it's the mucous neck cell. It tells you in the name, it has mucus in it. Mucous neck cells make mucus, but why? Why do you need mucus in the stomach? It's actually there to help protect the stomach, help protect from all this harsh acid. If you wondered how the stomach could break things down, not itself. All the mucus coming from the mucous neck cells creates a coating of mucus that can help neutralize any acid. But I keep it simple on your exam. A cell in the stomach is a mucous neck cell, and it makes mucus to help neutralize the acid for protection. Keep going. Another cell is the parietal cell. The parietal cell makes the acid in the stomach, and it's a strong acid. It's hydrochloric acid. Parietal cells make hydrochloric acid. Keep going. Turns out parietal cells also make intrinsic factor. Turns out intrinsic factor is required for us to absorb vitamin B12. When you eat food that has vitamin B12 in it, it will make its way all the way to the intestines. It will not be absorbed unless you have intrinsic factor coming from the parietal cells. All right. Keep going. There's the chief cell. I mentioned this in your exam review. Chief cells make the enzyme pepsinogen, and this is the inactive form. Pepsinogen will then get activated by all the hydrochloric acid in the stomach into its active form, which is pepsin. And it's pepsin down proteins into amino acids. All right. And then one last cell. It's called the G cell, sometimes called the enteroendocrine cell, or sometimes called DNES cells. All right, lots of names. This cell, if you call it a G cell, it gives you a hint. G is for gastrin, which is a hormone. That's what these cells make. And what does gastrin do? Gastrin actually goes back to the stomach, tells the stomach cells to produce their products. So it's really a chain of reaction that occurs. You see, smell, look at, or even taste food. These cells begin to pump out gastrin, which will stimulate the other cells to secrete their products. Parietal cells to secrete hydrochloric acid and intrinsic factor. Chief cells to release pepsinogen. And mucous neck cells to release mucus. So those are the cells. That's what they make and what they make do. G cells make gastrin. Gastrin stimulates all the other cells in the stomach to release their products. Chief cells make pepsinogen, which gets activated to pepsin, which breaks down proteins. Parietal cells make hydrochloric acid, which can break down food, and it makes intrinsic factor to help vitamin B12. And mucous neck cells make mucus to help neutralize all the acid in the stomach for protection. So those are cells. And then you got to know the three parts of the large intestine in order. We saw that it's in lab. It's duodenum, jejunum, and the last part is the ileum. We know from lab some things we'll see when we look at these three parts of the small and large intestines in under the microscope. We know when we look at small intestines, duodenum, jejunum, and ileum, we will see villi, mucosa. We'll see that brush border, the microvilli. If you just cut open the intestines, both small and large, you'll see this though. You'll see circular folds. All right, let's go to a picture so I can show you circular folds. Go down to intestines. Oh, here we go. When you cut open the intestines, it's not smooth on the inside. You have these like ridges. They're just projections of the mucosa. These are called circular folds. Oh, they look like these are not villi. The villi are these little spikes, if you were to zoom in, kind of sticking out. All right, so these are like huge humps, and then you have the villi sticking out. Why do we have all these things? Why do we have circular folds, villi, and microvilli? They all help to increase the surface area to allow for more absorption. The major function of small intestines is to do absorption. So they're going to have lots of things to increase their surface area so that they could do lots of absorption. And what things? Circular folds, villi, and microvilli. Keep going. Remember when we looked at the duodenum, we saw things like Brunner's glands and the submucosa. And when we looked at ileum, we saw things like Peyer's patches helping to fight infections. And you remember the jejunum really didn't have any other special features. And when we looked at the large intestine, when we looked at colon, remember it didn't have any of those villi or microvilli. All right. And lacteals. We saw lacteal in the lab practical 2 review when we looked at the model of the intestines. And you saw these finger-like projections with these little tubes inside. And I pointed to the tube inside and called that a lacteal. It's the blood vessels inside of a villus. And their job is to absorb nutrients. When we talk about absorbing nutrients, yes, the cells take it in, but they kick it into the lacteals. So it's the lacteals that are really helping us to absorb nutrients. And then we do the same thing that we did with the stomach for the small intestines. Just how the stomach had cells that made products that help to break food down. Well, your small intestine also has cells that will also make products to help us out. So let's go on over to the cells of the small intestines. So I'm just looking for the slide. Okay. Sorry, this is going to have to be the slide. So this is talking about some hormones and where they secrete from, where they going, and what are the effects they're doing. And we're just talking about the intestines right now, mainly talking about the duodenum. Well, duodenum, you can see from our list, makes 2 major hormones and 1 major peptide enzyme. All right, there's cholecystokinin, secretin, and VIP. VIP stands for vasoactive intestinal peptide. These are all coming from your small intestines, mainly the duodenum. And you got to know what these do. And we're going to start off with the hormones cholecystokinin and secretin. It helps to know where they're coming from because it gives you a hint. They're both coming from the duodenum. But I think the duodenum is the first part of the small intestines right after the stomach. stomach empties all its products, the duodenum is going to get it first. And when the stomach empties, it empties everything, including the acid inside. So in addition to having to help to absorb nutrients, it has to protect itself from all the acid coming from the stomach. Turns out it will get help by releasing a hormone. Turns out it will get help by releasing cholecystokinin, and it will get some help by releasing secretin. In addition to having to protect itself from acid, when the stomach empties, stomach doesn't always break down. Well, small intestines doesn't food down. You saw its major function is absorption. So if it wants to break food down, it's going to have to call for help with these hormones. So which of these hormones is helping to protect it from acid, and which of these hormones are helping it to break down partially undigested food that came from the stomach? Well, it's all here on this slide. All right. For one, there's cholecystokinin. For one, cholecystokinin forces the gallbladder to release bile. And remember, bile helps to break down fat. So one function of cholecystokinin is to help break down undigested food, just in case stomach released something before it was fully digested. Duodenum releases cholecystokinin, which will trigger the accessory organ, the gallbladder, to help release bile to help finish off the digestion. All right. In addition to causing the gallbladder to release bile, it also causes enzyme to release the pancreas, to release its enzymes as well. Again, to help finish digesting those undigested foods coming from the stomach. And then it needs to protect itself from acid too. Turns out it needs to ask for help again from accessory organs. Again, it's asking for help from the pancreas, this time with secretin. Secretin causes the pancreas to release bicarbonate. Same thing you have in your saliva that can neutralize acids. So this is why you're also not burning up or eating up your intestines with acid, because they also neutralize the acid, but they're doing it with help. Okay. So that's all these hormones are doing. Going to other organs so that they release products to help the duodenum. All right. So think of cholecystokinin and secretin as the duodenum asking for help. It releases cholecystokinin, it's asking for help to finish breaking down undigested food. So cholecystokinin will trigger the release of bile from the gallbladder and enzymes for the pancreas to help do that. While secretin will help to protect it from all the acid. How? It will go to the pancreas to release bicarbonate. And then there's VIP, stands for vasoactive intestinal peptide. It's a peptide, so it's a protein, so it's an enzyme really. And it turns out this will actually provide some protection also from acid, but it's major thing here. Relates the pancreas again to release its products, mainly enzymes. So again, it's helping to break food down, kind of very similar to cholecystokinin. So those are really the products of the small intestine. Don't worry about the cells themselves. There will not be a question asking what are the cells of the small intestine. Just know those major products: cholecystokinin, secretin, and VIP. All right. So I know I wrote lysozyme, which kills bacteria. We know that's coming from our saliva, helping us to kill bacteria. So we talked about cholecystokinin and secretin. So then we move on to the large intestines. The major function of the large intestines is to absorb water, in addition to absorbing some vitamins, and also it's responsible for defecation, helping us to get the feces out of the body. But there's some special structures on the large intestines that need some explanation. One are what we call tinea coli. Tinea coli are little bits of muscle. Let me go show you a picture of large intestines. Here we go. Here's a picture of the large intestines. Remember, it's large, so we break it up. It begins with the cecum. Remember, you could always identify the cecum because it attaches to the small intestine. It attaches to the ileum of the small intestines and the appendix in most people. Then after the cecum, you climb up the right side of the abdomen as ascending colon till you hit the liver and you bend at the hepatic flexure. Then you cross from right to left as the transverse colon until you bump into the spleen, and then you curve the left side of the body as the descending colon. So you hit the pelvic floor curving as the sigmoid colon. Then you finish off as the rectum and anus. And on this picture, you see this little white line kind of traveling the length of the large intestines. That is your tinea coli. It's muscle. Okay? And because it's muscle, it gives you a hint as to its function. Muscles move things. Tinea coli are also helping with peristalsis, with that propulsion of food through the large intestines. It's helping with propulsion, moving food along. Talked about the subdivisions of the large and small intestine. In order, it's duodenum, jejunum, ileum, cecum, ascending colon, transverse colon, descending colon, sigmoid colon, rectum, and anus. And I mentioned one of your last sphincters are the internal and external anal sphincters between the anus and external environment. You got to know which one do you control, which one is voluntary and which one is involuntary. Now, let me give you a hint. Internal anal sphincter is made up of smooth muscle, while the external anal sphincter is made up of skeletal muscle. So think back to your tube. You know which one you control. Turns out you control the external anal sphincter. It's voluntary because it's skeletal muscle. Remember, we control skeletal muscle. But your internal anal sphincter, oh, that's smooth muscle. So it's involuntary. You do not control the internal anal sphincter. And that's a good thing. We see a picture of the sphincters. Internal anal sphincter look more internally closer to the anal canal, while kind of lower down and further out, you see the external. It's lower down. Externals are the last sphincters. So you can imagine, oh, you're the one responsible for holding the door. If your pants. Uh-oh, you can't blame anyone but yourself because you control the last door. Uh-oh. Okay. But remember, keep it simple. What do you got to know? You control the externals. It's voluntary. You do not control the internals. It's involuntary. Remember, you got to know the major functions of these organs. Remember, small intestines deals mainly with absorption of nutrients, while large intestines deals mainly with the absorption of water and some vitamins. And then when I told you, we've got to talk about pooping. Okay. Remember, pooping is called defecation. We're not going to go into the detail. I just want you to know what triggers pooping. Oh, you might think it's you, but no, it's not. How do you know? Just ask anyone. Poop activate. Oh, it doesn't work like that. Okay. You can't force it on command. So something controls it for you. All right. It's your body. It's part of your nervous system that you don't control. What tells your body you have to poop? What's the trigger for your defecation reflex? What's the trigger for pooping? How does your body know you have poop to get rid of? Well, something needs to fill up. It's your rectum. Turns out when your rectum fills up, it stretches. That stretching is sensed by receptors. Oh, that sounds a lot like some feedback mechanism where a receptor notices, tells a control center, which comes up with a plan, and the effector put it into motion. Yes, your pooping is a reflex. All right. And it's triggered by stretching of the rectum. That's the major indicator that your body knows that you have to poop. And that's all this is talking about in your exam review. It begins with stretching in the rectum, and that stretching is picked up by receptors. And that's how your body will know you're ready to defecate. And it will trigger the. So it turns out, no, you don't control pooping. Your rectum does. Keep going. Oh, not only do you have poop in your intestines, you have bacteria. People talk about poop being nasty and dirty. Well, technically it's true because you might have some bacteria in your poo because you have bacteria in your intestines. There are bacteria that live in your intestines normally. We call them your normal flora. All right. And they have function. You have a symbiotic relationship from them. They live in your intestines for shelter and food, but then you get things in return. Like what? Turns out these bacteria could produce some vitamins. They can produce some vitamins your body can't. So that's one reason why we want these bacteria around. Keep going. In addition to providing some vitamins, turns out they help to deter the growth of very harmful bacteria. Having your normal flora living in your intestines helps to inhibit harmful bacteria from taking root. So that's another function of having a normal flora. Keep going. You got to know the major compound produced by the liver and its function. That's bile. And I told you bile breaks down fat. To emulsify fats means to break down. And we, like always, give credit to organs and glands, but it's really the cells. There's a cell in the liver that's making the bile. It's actually liver cells. We call them hepatocytes. This is just, you got to know, hepatocytes are the cells of the liver, and they are the cells making the bile. And then we're finishing off with your accessory organs. Liver with its hepatocytes and bile to break down fats is one major function. And there are other organs. I mentioned them. It's things like your gallbladder and pancreas. These are your major accessory organs. And they're called accessory organs because it's like you have extra things, like an accessory. They're not part of the alimentary canal. They just contribute to it. So that's the liver, gallbladder, and pancreas. And I mentioned they're making products to deliver to the alimentary canal. So we want to know, how do you deliver them? How do you drain the products from the liver, gallbladder, and pancreas? Via ducts. You got to know the ducts. For example, draining just the gallbladder is the cystic duct. Draining just the liver or your right hepatic ducts, they merge together to form your common hepatic duct. So the common hepatic duct drains just the liver. But then there's a duct that drains both the liver and gallbladder. It's a duct made from the merger of the cystic duct and the common hepatic duct. It's called the common bile duct. So those are some ducts that drain your liver and gallbladder. But you also have to drain the pancreas. I'm sorry, I'm looking for slides while I'm talking in case nothing's wrong with your computer. I'm flipping while I'm talking. But also draining the pancreas is your main pancreatic duct. All right, and your main pancreatic duct will combine with the bile duct at your duodenal ampulla to release the product. Sorry, I was looking for this picture. All right. So I mentioned your common bile duct will merge with your main pancreatic duct to get released via the duodenum, via an ampulla and your major duodenal papilla. So one more time we go back. So we could see it on the picture. Just the liver, common hepatic duct. Just the gallbladder, cystic duct. And your cystic duct and common hepatic duct merge to form the common bile duct. And your common bile duct will make its way down in the abdomen and combine with the main pancreatic duct from the pancreas to get released into the duodenum. All right. And then we look at the pancreas. We know pancreas is both an endocrine gland and an exocrine gland because the pancreas release hormones which get released directly into the bloodstream, which makes it an endocrine gland. But it also releases enzymes that it up to release them. So it's also an exocrine gland. So we got to know, what are the exocrine products, what are the enzymes the pancreas releases, and what are the endocrine products, what are the hormones the pancreas releases, and what are these things doing. Let's start off with the exocrine. What are the enzymes and other chemicals the pancreas makes? We've mentioned some already. For example, we mentioned the pancreas releases bicarbonate to neutralize acid. Remember, secretin coming from the duodenum goes to the pancreas to tell the pancreas to release bicarbonate to neutralize. All right. So that's one product in its function. It's bicarbonate. What does it do? Neutralize acids. Remember, cholecystokinin and secretin are coming from the duodenum to go to the pancreas. All right. What else? Let's mention some hormones, some endocrine functions of the pancreas. Turns out the pancreas releases hormones via what we call islet cells. When you look at the pancreas underneath the microscope, come on down. Oh, it doesn't have it. All right. When we talk about the pancreas, it has what we call islet cells in areas we call islets or Langerhans. And these islet cells are doing the endocrine functions. The islet cells, they're making the hormones. And it turns out the pancreas makes two major hormones, insulin and glucagon. They kind of talk about it more in chapter 23. That's why it's not here. But now, since we're here, insulin, as its function, helps to lower your blood glucose levels. How? By telling cells to let glucose in, by stimulating glucose uptake of cells. Okay. So that's the major function of insulin. Insulin lowers blood glucose levels. How? By telling cells to let it in. You can imagine, if you're telling cells to let glucose in, it's taking it in from your bloodstream. So if I'm moving it from the bloodstream, it's going to look like its levels are dropping. So we say insulin lowers your blood glucose level. That's why diabetics need to check their insulin levels. They need to get shots depending on the type of insulin they have, because it will help to drop their glucose levels. Because with diabetes, your insulin or your glucose levels are too high. And then there's a different hormone called glucagon. Glucagon helps to raise your glucose levels. How? For one, it can help to stimulate the breakdown of glycogen. Oh, you got to think back to chapter 2. Remember, glycogen is a storage compound made up of glucose. So if you break it down, you'll get more glucose and your glucose levels will rise. So insulin and glucagon have opposite effects when it comes to glucose. It drops your glucose levels, while glucagon raises your glucose levels. I kind of mentioned diabetes. Diabetes is a disorder of an elevated blood glucose level. And that is diabetes mellitus. It's called sometimes blood sugar diabetes. It's because your blood sugar levels, your glucose levels are high. And this could be either from a lack of insulin. This is in type 1 diabetes, sometimes called juvenile diabetes, where they've destroyed their islet cells, which is where you destroy the place where you need to make it, well, you're not going to have it. And if you don't have insulin, you don't have something to drop your glucose level, so it stays high, and you have diabetes. But another reason why you might have diabetes is because of a lack of response to insulin. It's called insulin resistance. It's literally your body not listening to insulin. They still make insulin. Insulin still tells the cells to let glucose in, but it's like the cells say, "Nah, I'm okay." And because the cells let the glucose in, well, your glucose stays in your bloodstream. You have a high blood sugar level, and we call it diabetes. And then last thing for chapter 22, the pathway of nutrient flow. I wrote it in parentheses just to make sure. When you absorb your nutrients, remember you're going to drop it into the blood vessels in your lacteals. Those are veins. Right? And your veins will bring it to your vein in the liver called the portal vein. And that portal vein goes to the liver. And the liver is where you'll process your nutrients. Whenever you eat food, you don't technically use it right away, depending on what that nutrient is. You have to bring it to the liver for the liver to process it. Think of the liver as a big filter. It removes things, it activates things so that you could use nutrients. That's the pathway. Portal veins to the liver. And that's chapter 22. And then in chapter 23, we hit more of the metabolism because this is metabolism. We hit more of what happens after you eat and absorb your nutrients. After you drop your nutrients in the bloodstream, what happens? Okay. And like always, you know this stuff. You just don't know the fancy names. We know when we eat a Whataburger cheeseburger, we're going to have to break that burger down. Yeah. And we'll send it to the stomach where the stomach will break it down some more. You do a lot of breaking things down. You even do it at the chemical level. Remember, your stomach releases enzymes and a whole bunch of chemicals to chemically break food down. So we got to talk about reactions again. To me, chapter 23 sounds a lot like the second half of chapter 2. Sounds a lot like the biochemistry we talked about in chapter 2. So it helps possibly to kind of revisit some chapter 22. It would help to make chapter 23 make more sense. For example, some definitions are exactly the same from chapter 22. We're focusing on the chemical breakdown, chemistry. Remember when we talked about chemical reactions where you break things down, we gave a name to it. Remember that was catabolism. That was you breaking things down. What else? If we could break things down, we could build things up. You build large complexes from smaller subcomponents. When you build large things from small building blocks, that's anabolism or anabolic reaction. What else? Remember, when you're breaking and forming bonds, when you're breaking things down with catabolism or making things with anabolism, that involves making and breaking chemical bonds. And remember, you have energy in bonds. So sometimes when you break something down, you're going to break chemical bonds and release energy. So some chemical reactions will release energy. And we call those exergonic reactions. So that definition again. Remember, catabolic reactions are types of exergonic actions because they release energy when you break those bonds. Remember, when you make bond, that absorbs energy. So anabolic reactions tend to be endergonic because they need to absorb energy in order to form bonds to build your larger products. So if you notice, a lot of this is just us repeating chapter 22, especially about the basic definitions. All right. And we'll be mentioning these terms as we go through the rest of chapter 23. For example, breaking down sugar. The process of breaking down sugar is called glycolysis. Remember, I'm not going to go into this in detail. Remember, you do not need to know your individual chemical reactions. All right. So I'm going to kind of focus on these little notes I wrote down here, kind of walk you through the process of breaking down a sugar, breaking down fat, and breaking down an amino acid. So let's go chapter 23. All right. I'm going down to the section where they talk about glucose catabolism. All right, here we go. So you bite into your Whataburger cheeseburger. You're going to begin to break down the bun, which is carbs, with the salivary amylase. You're going to swallow it, send it to the stomach, where more enzymes and acids will break it down, and you will absorb it. And your body will break it down into its individual building blocks, the monomers. Remember, those are glucose. Focus on what happens to that individual sugar. What happens to the glucose when you're breaking down and absorbing nutrients? How are you going to use this glucose? For example, energy. And that's what we're talking about. Well, it happens in a series of reactions. Turns out it's multiple reactions. In order, I wrote it in your exam review. It's a glycolysis, followed by pyruvate phosphorylation, then the citric acid cycle, and then finish off with the electron transport chain and chemiosmosis. These are all going to result in the formation of lots of ATP. So you use glucose primarily for energy. And it helps to know where these things occur. I mentioned the first thing you're going to do is glycolysis. That's occurring in the plain old cytoplasm. Then you'll do some pyruvate phosphorylation and make acetyl-CoA, which will enter mitochondria into the middle. We call that the mitochondrial matrix, where you'll do your citric acid cycle. And you'll make some ATP plus some electron carriers. And your electron carriers will go on over to the electron transport chain, still in mitochondria, where they'll make some more ATP. So you got to focus on what do you start off with and what do you end up with in each of these little steps. And luckily, your textbook has some summary tables and some summary charts we could look at. Go into the summary chart for glucose catabolism. There it is. All right. So here's a little summary chart. What happens when you have one glucose molecule that you break down? First step is you're going to go through glycolysis. And in glycolysis, you're going to take glucose, which is 6 carbon sugar, meaning when you look at its chemical formula, you will see 6 carbons in it. And you're going to break it down into 2 3-carbon compounds called pyruvate. Okay. And then that pyruvate needs to go on. Actually, I'm going to show you. I think this might actually help. Glycolysis, we start off with glucose, and we're going to end up breaking it down into 2 pyruvates. And in the process of breaking it down into 2 pyruvates, we get some byproducts. We get some extra things. For one, we're going to make 2 ATPs, net ATPs. You make a total of 4, but we say you have a net of 2. Why? Let me go show you in a picture. Turns out when you're doing glycolysis to break down glucose into pyruvates, you need some energy to make energy. It's like you got to invest before you get a return. And investment is 2 ATP. So even though you make a total of 4 ATP after glycolysis, you got to refund. You got to kind of think you got to refund 2 of your ATPs. Here's the picture I'm talking about. Here we start off with glucose, which has 6 carbons, and we eventually end up with 2 pyruvates, each with 3 carbons. And we make a total of 1, 2, 3, 4 ATPs. But if you look at these chemical reactions, you notice way in the beginning, you had to use 2 ATPs. So when we talk of glycolysis, they love to say these 2 ways. Say you make a total of 4 ATPs or net of 2 ATPs because you had to use 2. Okay. And then you're going to have some electron carriers. When you're making and breaking these bonds, you move electrons around. And sometimes you're going to have loose electrons. You're going to need something to carry them. We call them electron carriers. They're NAD, which is the oxidized form, meaning without the electron, and NADH is the reduced form, meaning it has the electron. It's holding on to an electron. And there's FeD. That's another electron. FAD is when it's not carrying an electron, it's oxidized form. And FADH2 when it is carrying an electron, when it is reduced, it does have electrons. So by the end of glycolysis, you would have reduced 2 NADHs into NADH2. But keep it as simple. Keep it as simple as all. In glycolysis, you start off with glucose, and you end up with 2 pyruvates, 2 NADHs, and a net of 2 ATPs. And then pyruvate is going to move into the next step. You're going to take your pyruvates, and you're going to move into phosphorylation. So you know the beginning products. It's the 2 pyruvates you ended off with glycolysis. And those 2 pyruvates be phosphorylated to produce 2 acetyl-CoAs. All right. So kind of thing. Each pyruvate will get turned into an acetyl-CoA, and you'll have some extra carbon that will be blown off as carbon dioxide. And again, we're making and breaking bonds, so we'll have some loose electrons. Again, you're going to reduce some electron carriers. You're going to force them to hold on to electrons. Then keep going. It's just a chain, a sequence of events. We're going to take those two acetyl-CoAs and we're going to bring them to the citric acid cycle. You're going to have to do this cycle twice, once for each acetyl-CoA. So you start off with your two acetyl-CoAs. Then you're going to make your ATP. You're going to make and break bonds. So you're going to have to force electron carriers to carry electrons. And again, you'll have some leftover carbon. You'll breathe it out. Then by the end of it, you notice you've gotten rid of all your carbons now. And all you're really left with is energy and electron carriers. These electron carriers are going to finish off the game by going to the electron transport chain. So we're going to take all your electron carriers from all the previous reactions. So we have 2 NADHs from glycolysis plus 2 NADHs from pyruvate phosphorylation and 6 NADHs from citric acid cycle. So that's where we got the 10. And then we have our 2 FADHs from citric acid cycle. So we're going to take all these electron carriers and we're going to use them to help stimulate ATP synthase by driving our chemical or gradient of hydrogen to help stimulate and spin our ATP synthase to make ATP. I don't know what I talked about. Don't worry. It's not in the detail of this exam review. All right. We take our NADH, our FADH2, and we make ATP. So that's really what happens with glycolysis. That's all these slides are really going through. Start off with glucose, invest your 2 ATP, break it down into 2 pyruvates. You're going to have your NADHs holding onto your electrons, and you're going to make your net 2 ATP. Then you're going to take your pyruvate and you're going to do pyruvate phosphorylation, helping to make acetyl-CoA. And then that acetyl-CoA will then go ahead and enter your citric acid cycle, where each acetyl-CoA will go through a series of chemical reactions that will result in the formation of electron carriers, NADH and FADH2, as well as some ATP. Remember, you do not need to know this detail of chemical reactions. Keep it as simple as in your exam review. And then your NADH, your FADH2s will then go on to the electron transport chain, where they'll drop their electrons, which will drive hydrogen movement through the membrane, which will then come back down via its chemical gradient through ATP synthase, which will trigger the formation of ATP. And this makes a ton of ATP. So that's what happens with just one sugar molecule. And from one sugar molecule, you make a lot of ATP. And that most of that ATP is coming from the electron transport chain. That's glucose catabolism. Let's go back up now to the exam review. We got through glucose catabolism, so we can do a couple of these points. Okay. You got to know where these things are occurring. So we know how to break down sugar. It turns out you can make sugar. Okay, it's called gluconeogenesis. You make sugar. You got to know where. Turns out a major place where you make sugar, where gluconeogenesis occurs, is in the liver. In the liver. You actually make some sugar in the liver. Keep going. Not all of these are things you need to answer. Some of them are just things you need to know, like the next one. You just got to know that ATP hydrolysis, the breakdown of ATP for energy. Whenever you hear ATP hydrolysis, think you're breaking ATP down for energy. We talked about endergonic and exergonic reactions already. You just got to know that sugar, glucose, is the major energy source for the brain. But if sugar is not around, like in the event of starvation, sugar will use ketone bodies. We'll see this when we talk about fat breakdown. You got to know the overall chemical reaction formula for cellular respiration. Basically, you got to know the overall formula for breaking down sugar. keep going. We went through the steps. So we went through the steps of glucose breakdown already. Um, you talked about all this. I'm just reading through the exam review. I'm sorry. Okay, yeah. Then we're going to fat. So we kind of walked you through the steps of breaking down a single sugar molecule. Well, now we got to break down fats. And when we break down fats, we're breaking down the major fats in your body. The major fat for storage of energy in your body are triglycerides. Those are the major energy storing fats in your body. Okay? And so when we talk about breaking down fats for energy, we're going to break down a triglyceride into its building blocks and break those building blocks down for energy. And so it helps to know what are the building blocks of triglyceride. What are the major parts to a triglyceride? It's a glycerol molecule and 3 fatty acids. One triglyceride is made up of one glycerol molecule and 3 fatty acids. And so when we break the triglyceride, a fat, we break it down into glycerol. So we're going to have to see what happens to the glycerol. And when we break it down into the 3 fatty acids. So we're going to have to see what happens to the 3 fatty acids. When it comes to glycerol is pretty straightforward. Glycerol is a carbon-based structure. So you can really to wherever you need or have carbon-based structures. So think about where we just saw carbon. It was with our glucose breakdown. I told you break down your 6 carbon glucose into 3 carbon pyruvates. And turns out there are sub or kind of intermediate products. If we go back to our glucose metabolism overall breakdown, all those chemical reactions, you'll notice that one intermediary part or one chemical in that chain of reactions is glyceraldehyde 3-phosphate. Look at the name. Don't be intimidated. That's glyceraldehyde. That glycer and glycerol is because it was a result of glycerol. So when you break down a fat into its one glycerol and its 3 fatty acids, that glycerol could simply get put into our glycolysis reactions as glyceraldehyde phosphate. The 3 fatty acids will have their own journey. Okay. So let's go through this. First step, you eat your Whataburger cheeseburger. Well, that's cheese in it, and that cheese has fat. It has triglycerides. You're going to break it down into glycerol, and I mentioned glycerol will become glyceraldehyde 3-phosphate and finish off glycolysis, resulting in pyruvates. But then what happens to the fatty acids? Well, they will go on. They will go on to do what's called beta oxidation. This occurs to the fatty acids only. Okay, so you could think of it as fatty acid beta oxidation. So the starting material is going to be your fatty acid. And what are you going to break it down into? Oh, look at that. You're breaking it down into something familiar. You also break it down into acetyl-CoA. And yet again, you're making and breaking bonds. So you have free and loose electrons. So you have to force them onto your electron carriers. So we again get NADH and FADH2. You know the rest. You know what happens to acetyl. It's the same thing. It's still the same acetyl-CoA that we saw in our glucose breakdown. So where's that acetyl-CoA going to go? It's going to go to the citric acid cycle. You know the products of citric acid cycle. It's just that we only have one acetyl-CoA in this case, not 2. So this is the number you get. It's half the number in glucose. Remember in glycolysis, the sequence, you had 2 acetyl-CoAs. But here you only have 1. And again, you'll have your electron carriers that still again go onto the electron transport chain to again make ATP. All right. So here our body is using a lot of the same final pathway when it comes to making energy. It's using those citric acid cycle and the electron transport chain again. So again, this is all your PowerPoints are talking about. When you want to break down fats, you start off with the lipolysis, which is the process of breaking down fats into their glycerol and their three fatty acids. Then that glycerol will be converted into glyceraldehyde 3-phosphate, which will go on to make pyruvate and glycolysis and finish off that series of events. While the three fatty acids will go on to do beta oxidation. Oxidation, just again the process of breaking down now the fatty acids. And again, you know what you're going to break down into. You're going to break down into acetyl-CoA. And again, you're going to get some electron carriers. And then that acetyl-CoA will go on to the citric acid cycle, which will make some ATP plus more electron carriers. All right. So that's fatty acid oxidation. But we got to talk about this here. You have, depending on the situation, fatty acids may not undergo beta oxidation. They don't undergo beta oxidation, they'll get into ketone bodies. Ketogenesis is the process of making ketone bodies, and that occurs to some of your fatty acids. All right. So that's all this is talking about. That's fatty acid catabolism. And then the last thing we talk about is amino acids. You got to break down proteins. So when you break down your protein, you'll break down. Again, let's go to our exam review because I wrote it out in a little sheet here. So again, one last time, we bite into our Whataburger cheeseburger. We bite up the meat in the burger. We break it down into its proteins in our stomach, and we break the proteins down into amino acids, and we absorb the amino acids. What's going to happen to those? Well, turns out they're not quite ready for your body to use because they have something your body does not really use that much. It has what we call amino group or something containing nitrogen. Your body doesn't really use this nitrogen your amino acids. So when you have an amino acid, the first thing your body's going to have to do is break off the parts containing the nitrogen. We call that transamination. Transamination is just your body removing the nitrogen. That's all it is. Okay. Because remember, your body's stuck to the carbon. It wants the carbon. Because so in glycolysis and glucose catabolism and fatty acid catabolism, your body cares about the carbon for energy. So it doesn't care. It gets rid of the nitrogen as the very first step via transamination. And then it has the remaining carbon. And it's the remaining carbon it's going to use. Right. So you start off with your amino acid, remove the nitrogen, so you're left with the rest of the carbon skeleton of that compound. The nitrogen gets trapped as glutamate, and glutamate will undergo oxidative deamination to produce ammonia, and your body will get rid of ammonia in the urea cycle in your urine. Okay. When you talk about that urine smell, what you're usually smelling is ammonia. All right. And that ammonia is just an indicator of your body getting rid of that original nitrogen attached to your amino acid. But then what happens to the rest? After transamination, what do you do with that carbon skeleton? How do you get the energy out of it? That carbon skeleton gets turned into, guess what? Look at these things here. It could get turned into pyruvate, acetyl-CoA, or just straight up glucose via gluconeogenesis. We know what all these things do. If you turn that amino acid's carbon skeleton into pyruvate, it will do pyruvate phosphorylation to make acetyl-CoA, which will go to the citric acid cycle to make ATP, and electron carriers go to the electron transport chain to make more ATP. If you turn the carbon skeleton into acetyl-CoA will go to the citric acid cycle to make ATP and electron carriers, and the electron carriers will go to the electron transport chain to make more ATP. If you turn those amino acid carbon groups into glucose, glucose will go into glycolysis, which get broken down into pyruvate, which will undergo pyruvate phosphorylation to make acetyl-CoA, which will then go into this citric acid cycle to make ATP and electron carriers, and all your electron carriers will go to this electron transport chain to make ATP. You see, all these catabolism groups are all relying on the same steps: citric acid cycle, electron transport chain, and you will get energy. So that's all of this is really what this chapter focuses on. Just goes to the detail of all the little steps. But keep it as plain as and simple as it is in this exam review. All right. We went through that. We talked about insulin and glucagon. Insulin will drop your blood sugar levels while glucagon will raise it. So whenever you eat, you're bringing in glucose, so your insulin levels should go up when you're eating. And when you're not eating, your insulin levels should fall down because you don't have any glucose to bring in. When you're eating, we call that the feeding or eating state. When you're not eating, we call that the fasting or starvation state. So when you're not eating or starving or fasting, your blood sugar levels are low, so you'll release glucagon to raise it. And when you're eating, you release insulin to drop it. And then the last things we talk about, we kind of mentioned the citric acid cycle is a way of getting rid of your nitrogen in the form of ammonia and your urea. And then the last thing I want to hit are some lipoproteins. There are what we call LDLs and HDLs. If you ever donated blood or had your cholesterol checked, they always give you your cholesterol levels and your LDL and HDL levels. We're talking about fats, really. Okay. Turns out your body combines fats with some proteins to form a lipoprotein. Remember, don't be intimidated by names. It's telling you what it's made up of. It's a lipoprotein. So it's part fat, lipo is for lipids, which are fats, and part protein. But there are differences in their chemical composition. If you have very little fat, you're a low density lipoprotein, meaning you have very little protein and a lot of fat. While an HDL is a high density lipoprotein, meaning they have a high density of proteins and very little fat. Turns out you want very low LDL levels and you want high HDL levels because of what they kind of help do, mainly HDLs. HDLs, I told you, they tell you the name high density lipoproteins, meaning they are made up mainly of protein with a little fat. They have a high density of protein. And because of this, it could do things. Turns out a major function of HDLs is to transfer cholesterol to the liver so you could excrete it. Okay? Pump it out in bile so you could poop it out eventually. So HDLs can help to lower your cholesterol levels. That's why you want high HDLs. If your doctor ever tells you, "Oh, you got high HDL levels," you better snap back and say, "Oh, whoa, I better have very low cholesterol levels because my HDL is helping to get rid of it." LDLs do not help to get rid of it. Right? In fact, they're mainly fat. So HDLs in your body are like you having clogged arteries. So you want a low LDL level. Right? So that's the difference. There's good fats, HDLs, and bad ones. Right? LDL. Your exam review. Hopefully, I've gone through enough of it to help. Remember, if you still have questions, don't be afraid to ask. Don't be afraid to use the discussion board. I promise it is not stupid questions. I'm not getting a lot of good questions. I'm just getting a lot of the same good questions via email because you guys aren't seeing your questions and my responses. I promise people have your same question. Don't be afraid to use the discussion board. Otherwise, that's it. Don't be afraid. You can still email me, but don't be afraid to use the discussion board as well.